Hello, good afternoon, everyone. Please, this is just to let you know that we are prepared and ready to start in a few minutes while other participants are joining. So we kindly ask you to wait still some few minutes. Thank you. Hello, good afternoon, everyone. Thank you for joining our Avantor webinar today. My name is Maria Castaño, and I'm Life Science Global Campaign Manager at Avantor. I'm very pleased to welcome you all to this webinar, where you will learn about how to reduce failure risk due to faulty plasmids and speed up your discovery process. Next slide, please. I'm glad to introduce our speakers to you. Aaron Pomeranz, Segment Marketing Manager, Jenma Jordan, Product Management Director, and Stephen Rath, Associate Director for Bioinformatic Products, all from Oxford Nanopore Technologies. Next slide, please. Our speakers today will discuss about why the scientific community is quickly moving towards routine whole plasmid sequencing to reduce failure risk and speed up discovery process, how plasmid sequencing with Nanopore technology provides fast and comprehensive insight for plasmid verification, and the steps for the end-to-end plasmid workflow, including library preparation, sequencing, and analysis of complete constructs. Next slide, please. Before I hand over to our speakers, I would like to provide some important information about how the webinar will progress. After the presentation, there will be a Q&A session. please write and send your questions in the questions chat you see on your control panel. They will be answered in the Q&A session or directly by email if we would run out of time. Our guest from Oxford Nanopore will handle and answer the coming questions. This webinar will be recorded and you will get the link by email after the session, so you and your teams can go through it at your convenience and when you need it. At the end of the webinar, you will also receive a survey, and we would appreciate if you could please fill it. It's very important for us to get your feedback to improve the quality of our webinars. And finally, for additional comments or questions, you can always contact us on webinar@avantorsciences.com or visit our website at bwr.com. Thank you all and enjoy the presentation. Aaron, please welcome and you can start.
Good afternoon, everyone. My name is Aaron Pomerantz, and I'm the Segment Marketing Manager for Microbiology and Infectious Disease at Oxford Nanopore Technologies. Before I hand over to my colleagues, I would like to give a short introduction and overview of why the scientific community is rapidly adopting the use of nanopore sequencing for whole plasmid verification. Plasmids are typically small, circular DNA molecules that exist naturally in bacteria and have also been engineered to become ubiquitous tools in genetics and biotechnology labs today. Plasmid constructs are the backbone of molecular biology, serving as fundamental input material for various uses, from basic research to industrial applications, gene therapy, vaccine development, genetic engineering, and more. Plasmids have enabled fundamental studies into gene function, regulation, and expression, as well as for large-scale production of proteins of interest. Plasmids can also serve as vectors for gene manipulation, mRNA vaccines, and various applications that contribute to scientific advancements and industrial processes. But we know that errors can arise during plasmid assembly due to a variety of factors, such as imperfections introduced during enzymatic processes, as well as mechanisms involved in bacterial repair and replication. Because plasmid constructs are fundamentally important for downstream experiments and applications, scientists often employ various quality control measures to ensure the accuracy and integrity of their plasmids. For instance, Sanger sequencing has been widely used as a method to confirm accuracy of the DNA sequence. By comparing the generated sequence with the expected sequence, scientists can verify the correct assembly of a plasmid and identify any potential errors or mutations. However, Sanger sequencing has several limitations and undesirable features, such as a limited read length of about 1,000 bases, requirement of specific primers that bind to the template DNA during the sequencing reaction, and high relative costs when processing or outsourcing large numbers of samples. On the other hand, as we'll hear in a few moments in more detail from Gemma, nanopore sequencing overcomes such limitations, enabling direct, long-read sequencing of the entire plasmid, thus providing a comprehensive overview of its sequence, structure, and potential modifications. Legacy methods, such as Sanger sequencing, often target only a specific short region, limiting the scope of the analysis. On the other hand, nanopore sequencing enables the highly accurate, flexible, and secure characterization of full-length plasmids, with results that can even be obtained in hours. Using this method, the entire plasmid can be sequenced rapidly, locally, and at a competitive price. This enables several advantages over alternative sequencing methods. For example, users can easily identify if the plasmid insert and backbone are free of mutations, completely avoid the need for primers, complicated primer walking, and annealing issues, resolve repeat regions as well as dimers and deletions, and employ a technique that works across a wide range of plasmid and/or back sizes and complexities. This is also a simple end-to-end workflow with rapid turnaround time, and this process can be kept in-house or at your local institution, helping to ensure that your data remains secure. Whole plasmid verification with nanopore sequencing has quickly become a popular application within the scientific community. Here are just a few examples of what current users are saying, such as full vector sequencing has accelerated our cloning pipeline significantly, truly transformative. Could have never imagined having a fully annotated plasmid map for so cheap. Planning to sequence all of our constructs now. And in reference to plasmids used in gene therapy, total game changer. Sequencing through AAV ITR is no problem. And finally, no more test digests, no more colony PCRs, no more having to do multiple mini preps just to get enough material to primer walk through a large payload, no more fiddling with new Sanger primers. Lastly, let's take a look at a couple of recent case studies demonstrating the use of highly accurate and cost-effective nanopore-based plasmid validation approaches. In the top publication, the authors showcase a number of examples in which whole plasmid validation captured unexpected events that have functional consequences. For instance, unexpected deletions within promoters that would otherwise have been missed by alternative screening methods. We also see comparisons with the nanopore-based approach to Sanger sequencing-based approaches on the bottom, in which the proportion of the plasmid covered by nanopore assemblies are in orange and Sanger in blue, highlighting how nanopore assemblies spanned the entirety of the plasmids, while the alternative Sanger method covered a median of just 10.3% of their respective plasmids. So overall, what should a complete plasmid sequencing workflow look like? Ideally, it should be able to generate highly accurate sequence information for the entire plasmid construct, enable rapid, even same-day turnaround time at a competitive price, provide a simple point-and-click data analysis report, and facilitate sequencing at your local institution or in-house, ensuring that your data remains secure. Legacy DNA sequencing methods like Sanger typically only verify a partial region of the plasmid construct, which can miss important information and can result in failed experiments, unnecessary troubleshooting, and increased costs. On the other hand, as you'll hear in the remainder of this session, the nanopore-based workflow is easier, faster, and provides more complete information to verify your entire construct and ensure robust quality control. With that, I would now like to introduce my colleague, Gemma Jordan, who will walk us through the protocol for plasmid sequencing using Oxford Nanopore rapid barcoding kits.
Hi, I'm Gemma, and I'm here to talk you through the wet lab protocol for plasmid sequencing with Oxford Nanopore. So firstly, I just want to touch a little bit about the basics of Oxford Nanopore sequencing and how it works. So you can see here on the left that we have a blue nanopore that's embedded in a supporting membrane. We have DNA or RNA strands, which can be tethered down to this membrane and can then translocate through the nanopore. And the rate at which this translocation occurs is controlled by a motor enzyme, which is present in our nanopore sequencing kits. By controlling this rate of translocation, we can enforce a signal disturbance that occurs as the bases pass through the nanopore. And this electronic signal can then be turned into base calls or sequence data. And this is what's depicted on the right of the screen. There are many benefits or advantages to using nanopore sequencing. One of them is that we can do direct sequencing of DNA or RNA, and this provides more information for our users, so we can look at epigenetics or methylation or other modifications. It also allows us to do simpler workflows. We can enable workflows that are PCR-free for users that are not able to use that. We also can sequence a range of sequencing lengths, and we can provide data in real time, and this leads to rapid turnaround time in terms of results. We can also scale. We offer barcoding, we offer a range of sequencing device formats, so we can really accommodate any scale that a customer wants to achieve. And it's this unique combination of features that makes Oxford Nanopore a perfect choice for plasmid sequencing. Another great thing about the plasmid sequencing workflow from Oxford Nanopore is that it can slot directly into your current workflow. So you would still take your colonies, you still grow them overnight, you can still use your mini prep kit of choice to do your library purifications, and then you go straight into a rapid barcoding prep. So our rapid barcoding kit requires 50 nanogram per sample input. We can process up to 96 samples at once, and we can do this in as little as 60 minutes. Once you then have your library, you can sequence on a nanopore device. And for plasmid sequencing, we recommend the MinION flow cell, and this is able to generate data for 96 samples in approximately 2 hours. We then offer analysis solutions, so we have our Epitome software, which then enables you in less than an hour to generate consensus genomes for all of your plasmids. If you'd like to view the full protocol, it is available on our Nanopore community. So going into a little bit of detail now about how our rapid barcoding kit actually works. As I mentioned, the input is the DNA that you've gotten out of your mini prep kit or plasma purification kit, of which there are a range available. And you take this and you slot it straight into our library prep kit. So we offer either 96 or 24 barcoding formulations of our rapid barcoding kit, but they both work by the same method. So we cleave the DNA by the use of a barcoded transposome complex. So not only does this cut the DNA, but it also adds a pair of barcodes onto the ends of your DNA fragments. As all of your samples are now barcoded, they can then be pooled together and cleaned up in a single bead cleanup step. So this makes the workflow incredibly rapid. We can then attach our sequencing adapters prior to loading onto a Nanopore sequencing flow cell. And there are very limited third-party reagents required for use of our rapid barcoding kits. So we do also offer a flow cell wash kit, which can be used to maximize higher sample throughput. And it works by using a nuclease treatment to remove DNA from the flow cell and enable the flow cell to be in a state where it can be reused. One thing I'd like to point out here as well is that unlike traditional methods, you don't need to order any primers for plasmid sequencing with Oxford Nanopore. We don't target a specific area of the plasmid. Instead, we are targeting the entire plasmid, if you will. And that means that there's no extra third-party primers that you need to buy or validate prior to utilising this method. So now I'd like to pass over to my colleague Stephen Rudd, who's going to talk you through the analysis solutions for Oxford Nanopore plasmid sequencing. Thank you.
So fabulous. Thanks for the great introduction, Gemma. So now that we have a collection of DNA sequences, we need to have a little bit of a think about how we're actually going to process the data. What are we going to do? How can we recover a plasmid sequence? So this schematic is really showing our, the basics of our recommended approach to solving and recovering complete plasmid sequences. So the way that we're going to validate our clone sequences is with what we call WF clone validation, and this is our supported workflow for plasmid sequencing, and it's part of our Epitome product line. So let's have a look in a little bit more detail what Epitome really is. So Epitome is our desktop application that provides Oxford Nanopore sequencing users with a collection of bioinformatics workflows that can either be run from the desktop or through the cloud. Of course, plasmid sequencing is not the most challenging bioinformatics workflow, so in this case it's entirely appropriate to imagine that this is a desktop-only type workflow. So let's have a little bit of a dive into, what are we actually doing? How are these workflows running? What is the workflow? So our WF clone validation workflow is implemented as a Nextflow workflow. Nextflow is a domain-specific language, and this now enables us to write multi-step bioinformatics workflows. It includes information that you provide, such as where is the primary sequence data found. It'll consume other sort of information as to how large do you expect your plasmid to be? What sort of QC are you interested in? Do you have an expectation as to what you expect to find within your plasmid? Tell me all about your primer sequences. So anyhow, Nextflow, domain-specific language, and WF clone validation is our workflow for looking at plasmid sequences. The reason that we've packaged everything into a workflow is to promote reproducibility, and I would say that really this workflow, although entirely functional, is also a fairly cool template that you as a user are welcome to fork, modify and adapt to the requirements of your own particular projects. So workflow as it stands provides a set of functionality. These are listed on the right hand side of this slide. So typically what the workflow will do is it will consume the folders of multiplexed or demultiplexed sequence data, and it's going to filter the sequences by length. The reason that we filter sequences by length is to avoid the construction of plasmid consensus sequences that are based on some of the sort of concatenamers that are often seen. So we're going to restrict the sort of size of the plasmids to a particular universe, keeps it clean, keeps it fast. The next sort of thing that we would also do is we would remove any of our host sequences. So typically plasmids will be propagated in an E. coli cell line. What we want to do here is we want to remove all of the E. coli sequence reads. There's always going to be a small amount, or in some cases a slightly larger amount of host contamination. So let's remove all of these sequence reads. If you're cloning from a sort of an E. coli, if you're expecting our inserted sequence to be of bacterial origin, then there are ways that we can go and either completely turn this off, or we can specify that actually you're working with a particular region of the genome that we should not be masking. Once we've got the sequences cleaned, once we've sanity checked a particular sort of library, what we do is we work through the assembly of the plasmid. The assembly is based on a couple of open source tools. Tricycler is a great piece of software for the recovery of bacterial and plasmid genomes. And what this will do is it'll subsample your sequence collection, and then we use the fly assembler to sort of perform some independent assemblies from these subsampled sequence collections, and Tricycler will then merge all of these sort of output together. It'll prepare a single consensus sequence, and this is the result. Of course, there's more we can do with the consensus. We do a little bit of polishing using the Medaka software, so this corrects some of the residual error within the assembly, and then we perform annotation with Planetate. Planetate is a really nice piece of software. It produces good looking graphs, it produces a nice plot, and it will also give a little bit of information as to what are the genes that are observed within the plasmid, what are the synthetic sequences, what are the primers, what are the adapters, what is really within your plasmid sequence. So the final thing we do is we extract what is the actual cloned insert sequence. This is based upon the presence of the primers, and then with this we prepare an HTML format report, and essentially that's it. We have a beautiful looking report and a good collection of data for sort of subsequent review. So how does it actually work? What is the actual process looking like? So this slide is just showing how do we actually start this through the Epitome software. So once you've installed the clone validation software, or once you've installed the workflow, then it provides you with a fairly clean dialog through which all you really need to provide is the path to the FASTQ sequence data. You should provide the software with a little bit of an estimation as to how large you expect your plasmid to be. So in this case, you can see that I've specified that we're expecting the plasmid assembly to be about 7,000 nucleotides in length. There are all sorts of other customizations that you can do, but the only sort of additional customization or adaptation to the workflow included here is I've specified that the number of CPU cores is 12. This just makes the analysis a little bit faster because it's going to use more of the resources that are available on this Mac computer in this instance. The cartoon on the top right of the screen shows what is the actual starting sequence data here. What is it that I am running the analysis with here? So here you can see it's a 12-plex analysis that Gemma produced a little earlier. So we just want to see, can we recover DNA sequences, or can we recover single closed plasmid sequences from each of these barcodes? So now, you click the Launch Workflow button that you can see at the top right of the main screen, and then you wait for a few minutes, and then it should produce a collection of results. So the highlight, the main result that we're going to consider here is the HTML report. The HTML report will present a certain amount of information for each of the multiplexed samples. This is shown in the main panel of the screen here. We can see that it produces a little bit of an infographic which reveals the number of reads. The number of reads can be switched between the number of reads that were observed and the number of reads which were actually used in the plasmid assembly. So in this case, we can see that for this barcode number 5, there were just less than 10,000 starting reads, but by the time we had performed the downsample, the number of reads that we selected was actually only sort of 250 sequence reads. This shows that for a successful recovery of a plasmid sequence, we do not really require thousands or tens of thousands of sequence reads. A modest number of reads is sufficient. In addition to the infographic, we present tables of information. We also present a number of plots that show the quality characteristics and the length characteristics of a sequence collection. It's always worth having a quick look at this. It just builds reassurance and it helps you understand, that everything is right. The planetate results are shown as the figure. Here we can see that we've got a circularized plasmid sequence of 3.0 kilobases of sequence. We can see the clone sequence, we can see the antibiotic gene, we can see the location of the primers and the particular origin of replication. So hey, this is an entirely satisfactory looking plasmid sequence. So this is, a successful plasmid assembly. To the right-hand side of the screen, we can see that there's a little bit of other information. We're going to present for each of the barcodes that are observed within a sequence set, what is the success or what is the end state of the analysis. In this case, we can see that they all completed successfully, and we can see the length of the circularized plasmid. So there's a little bit of variability here. We've looked at a number of different plasmids, some of which are replicates, and hey, there's numbers. It entirely works. This is exactly what we were expecting to see. The table at the bottom of the screen, where it presents insert sequences, gives you a little bit more information as to what was the length of the cloned insert that was observed within the plasmid, and between which primers was this observed. So this is, again, just a little bit more information. You have an expectation when sequencing a plasmid that you have a particular insert. This just allows a very quick eyeball analysis to see, have we recovered the expected information or not? So hey, HTML report is a great starting place to begin to question, are you happy with the way that your data has assembled? So really, that's all there is to the Plasmid workflow. It is a single workflow that really automatically just takes a collection of FASTQ sequence data and helps you towards the results. The results aren't just within the HTML format file. We also generate a folder of output. The output includes the complete plasmid sequence, but it also contains the insert sequence. We also present a bed file that describes the location of each of the genes and features observed within the plasmid. So hey, this is a great starting place. This is actionable information that can be used to question, is your plasmid exactly as you expected? I ran this analysis. It takes about 5 minutes to run this 12-plex dataset. If you need to have, answers a little bit quicker, or if you're running 96-plex and you don't want to wait, perhaps, 45 minutes for the complete analysis, then it is entirely possible to run this workflow entirely on a server. If you run it on a server with 100 cores or 120 cores, then you can run the same sort of 12-plex analysis in perhaps just 2 minutes. It's going to only assembly is the limiting step, but you're able to run a 96-plex in that same 2 minutes. So it's going to work very nicely, and you can really change your compute environment to suit what is the turnaround time that you expect from an analysis. If you have a more complex set of samples, where perhaps you've got some really large plasmids or perhaps some BACs, for example, then you should really be using a sample sheet, and the sample sheet will give you a little bit more customization as to how you're going to process each plasmid within a wider collection. These are functionalities that are available both through Epitome, the sort of desktop software, but all of this is also accessible through the command line. I would be recommending the command line where you're beginning to solve, data sets for a number of different customers, for a number of different users. Here you would use a sample sheet per customer, and you'd say that, for customer X, we should only be looking at barcodes 16, 17, and 18. And this way you can produce a single HTML format report per customer. So really, this is all there is to plasmid sequencing. Epitome is our solution for bioinformatics, and this is a product that is intended for you, our users. Please do let me know about more of your needs, and I'm going to be delighted to tell you in upcoming updates more about our functionality for sharing with you the sort of results and other workflows that will include, for example, our workflow for amplicon analysis. So with that, I'm going to say thank you so much.
Thank you, Aaron, Gemma, and Stephen, for this very interesting presentation. We will proceed now with the Q&A session. Questions will be handled and answered by Oxford Nanopore guests, Vishen Thackeray, Segment Marketing Manager, Jan, Senior Technical Application Scientist, Aaron Patel, Technical Application Scientist, and Sarah Griffiths, Bioinformatics Workflow Developer. Please enter your questions in the chat you have on your control panel, and then please, Vishen, we can start with the session and you can go through the questions.
Sorry, just getting off mute. Hello, everyone. Thank you for joining us for this webinar. I hope you enjoyed our session today. We've had a lot of great questions come in, so we're very excited to start answering some of your questions. Maybe we can begin with one of the first questions that came in. Is the E. coli genome sequence included in the software, or does it need to be provided by the user? I'll pass that to you, Sarah.
So it's not included in the analysis software. You have to input a host reference via the host reference parameter for whatever host reference you used for your workflow.
Thank you, Sarah. Let's move on to the next question. What kind of translocation occurs at the motor enzyme? Does the single-stranded DNA get pulled through the nanopore 3' to 5' or 5' to 3'? Aaron, would you like to take that one?
Yeah, I can definitely take that one. So the adapters are ligated to the 5' ends of the DNA molecule, so the translocation occurs from the 5' to 3' direction, and that's the same orientation in which the data is written.
Great, thank you. Let's go to the next question. Can I use this method to determine the complexity of a phage display library during my selection rounds? Obviously, the diversity is limited to a very short proportion of the plasmids, that is the single-stranded phage genome. Aaron, would you like to take this one? Can this be used to analyze phage display libraries?
Yeah, so our recommendation for phage display libraries and AAV type applications is similar, where we use the rapid barcoding kit for preparation of these samples and downstream sequencing. The analysis pipeline might be slightly different, but in terms of library prep standpoint, can definitely be used.
Perfect, thank you. Let's move on to the next question. Do we need a reference sequence for each plasmid? Sarah, would you like to take that one?
Yeah, sure. So the workflow uses de novo assembly, so you don't need to provide a reference for any of the assemblies, for any of the samples. You can optionally add an insert reference if you have one, and that will just be used for a few extra quality control steps in the analysis and will be output in the report. And that will be for across samples and across plasmids, so not per sample.
Perfect, thank you. Another question is, what's the difference between giving a reference for the workflow, clone validation workflow, or not giving one? Is it just better accuracy? I'll pass that to you again, Sarah.
Yeah, so as I just sort of just explained, the reference is only to do with the quality control steps at the end. So the assembly that is output will be compared to the reference, but only the insert section of the assembly. And yeah, it will just be used to do some extra QC steps, so it won't have any impact on the assembly itself.
Perfect, thank you. Aaron, the next one will be for you. What is the accuracy of the plasmid sequencing using this technology? Is it comparable to Sanger sequencing, or is it better?
In general, it's comparable to Sanger sequencing with much shorter turnaround times. And the advantage here is you can sequence longer stretches of the plasmid. The V14 chemistry combined with the R10.4.1 nanopore provides our highest raw read accuracy and can even produce duplex reads to further increase that accuracy. These duplex reads sort of combine data from both strands to base call into a single molecule at around Q30 or higher. And in fact, there are publications on our resource center that report median per base confidence of over 99.9% at a 10 times lower cost than traditional Sanger sequencing.
Perfect, thank you. And just to add to that question, because of the accuracy and quick turnaround time of whole plasmid sequencing using Nanopore, this is why it has so quickly been adopted by the biopharma and biotech industries, especially for processes such as developing and discovering antibodies or within the cell and gene therapy space. Moving on to the next question. Is the barcoding kit adding the barcodes randomly in the plasmid sequence? Aaron, would you like to take that?
Sure. Yeah. So the transposase activity does occur randomly, although it's at a finite amount of cuts per plasmid, and these number of cuts typically depend on DNA to transposase ratio and incubation time. The workflow itself will select the highest, best quality reads to use for the assembly. So number of cuts isn't really a limiting or an issue factor for this particular workflow.
Great, thank you. Maybe the next question we can take for you, Sarah. So when filtering by sequence length, what cutoffs do you use, and why do you use those cutoffs?
Yes, that's a good question. So we filter 0.5 times, between 0.5 times and 1.5 times the approximate size that was included. We do this so that there is plenty of data available for the assembler, and we're not filtering out any data that might potentially be useful for the assembly. The assembly will ultimately use a subsample of the best quality data available from that sample. But yes, we just don't want to filter out any useful data for the assembly. So that's why we give that wide range.
Great, thank you. The next question again is for you, Sarah. So does the downsampled reads used mean the rest of the reads were discarded? So for example, if there were from 9,000 raw reads, the downsampled was 200. Does this mean that 8,800 were discarded?
So yes, this is the case. So we choose the best reads available from the ones that there are, and often because the data is normally quite good, we don't need a huge amount of read depth to do the assembly. So yeah, so we about 200 is all that we take for the assembly.
Thank you. Aaron, this next one's for you. When the barcodes are added, is the plasmid converted to a linear molecule?
Yeah, so the transposase molecule will simultaneously add the barcodes and linearize any of the circular plasmids that are in the library, making it suitable for attaching the rapid adapters for sequencing.
Thank you. The next question is, what is the lowest threshold to identify mixed clones with single substitutions or indel mutations? Sarah, would you like to take that one?
So could you repeat the question?
Sure. What is the lowest threshold to identify mixed clones with single substitutions or indel mutations?
So there is a part of the workflow which looks for variants. For that, just the same coverage that you require for the rest of the workflow, and you'd have to input a reference insert, and that would give you some information on any variants that's found in the downsampled data. But in general, we don't search for contaminants in the data. The workflow doesn't, at the moment, cover that use case. Thank you.
The next one's for you, Aaron. Could you talk through the difference between the previous and new chemistry, that is the RBK110 versus the 114 kits?
Definitely. So the difference with the new chemistry is a different motor protein and pore chemistry. And this, as I mentioned earlier, this new chemistry provides our highest raw read accuracy and is duplex enabled, which means you can bioinformatically separate or identify your duplex reads, which are the template strand, the complement where the complement strand follows the template through the pore, and those can sort of be combined to provide base calls at a higher accuracy. So the main difference there is that you have new duplex enabled chemistry with our V14 kits, as well as existing features from our previous kits, such as fuel fix, which is not needing to add additional fuel during the sequencing run, and a higher capture tether to allow lower loading amounts.
And Aaron, what about the workflow? Can it be used, if you are not using the rapid barcode kit, can you still use a normal kit to do plasma sequencing?
Yeah, definitely. If you've linearized your sample sort of upstream of starting the library prep, those reads can definitely be taken into our workflow.
Thank you. The next question is for you, Jan. Can one use also the ligation sequencing kit and the native barcoding kits?
Yes. So yeah, related to what Aaron just answered, if you use ligation kit, you need to make sure to linearize the plasmid circular DNA first, so that it has double-stranded DNA ends to be ligate to the barcodes, and then sequencing adapter for sequencing. It will be a longer library prep compared to the rapid library prep protocol Gemma talked about. Yeah.
Perfect, thank you. The next question is for you, Sarah. What is the recommended sequencing depth or plasmid coverage to ensure a complete and high-quality sequencing using the Nanopore technology?
So it depends on the quality of the data that you have, but for coverage, I would probably suggest going for 60x coverage. That's what we have set as the default for the workflow. In terms of read depth, that needs to be a bit higher as we try to assemble the plasmid 3 times and compare the assemblies to get the best assembly. So for read depth, you would need maybe about, I'd say like at least 500, just to make sure that we can get enough good quality reads for the assembly to get that coverage of 60x.
Perfect, thank you. And the next question is related. Maybe you can take this one, Aaron. How many plasmids would you be able to sequence on a single fungal flow cell?
So as of right now, we don't have a published protocol for the fungal, although we do have a lot of community members that are sequencing on fungal flow cells to reduce the cost. Typically, our recommendation would be start with a fewer number of multiplex samples and then increase them once you realize you have enough coverage across each barcode. And there is a publication on our resource center that mentions a method for multiplex sequencing of a 16-plex library pool.
Great, thank you. Moving back to you, Sarah. How does the bioinformatic analysis distinguish sequences from different plasmids downstream or within the assembly step?
So unfortunately, at the moment, the workflow isn't set up to detect different contaminants. What probably would happen is you would get like an inconsistent assembly. And yeah, unfortunately, it's not at the moment set up to manage multiple plasmids in one sample.
Great, thank you. So maybe either Aaron or Sarah, both of you can contribute to this question. What is the best way to determine if my plasmid is a dimer? I guess that can be both a wet lab or bioinformatics solution.
Sure. Yeah, from a bioinformatics perspective, so you just do the assembly as normal, so do the workflow as normal. The sequencing should read all of the dimer as usual, and then when you run it through the workflow, you'll be looking for repeats in the assembly. So the way you can do that is by doing a self-alignment with the assembly that's output from the workflow. And we are currently adding something to the workflow in the next week or two, which should give some sort of analysis and visualization of any repeats in the assembly. So,
yeah, from a sequencing run standpoint, you might just want to look out for an N50 or a peak in your sequencing read distribution that's significantly higher, close to 2x times your expected plasmid length.
Thank you. And Aaron, would you be able to comment on whether we are releasing more than 96 barcodes, so can multiplexing be done with more than 96 samples?
As of right now, with the barcodes and indices we provide in the kit, the max is 96. You can design and sort of use your own custom barcodes, but this is a lot more work on the user end and would require you to sort of load Guppy with the config files for those respective barcodes. Our product teams are aware of the customer need for a multiplexing kit with a larger number of indices. So this is in the works, but no definitive timeline as of yet.
Thank you. Aaron, maybe you can support with this question as well. What is the maximum size of plasmid that you can sequence? What will happen if the plasmid is too large?
Yeah. So we do have some community members that are sequencing very large BACs, for instance, near the 200 kb range, as well as publications that report successful assembly of BACs. The sequencing itself may require longer run times and wash or reloads of the flow cell to obtain sufficient high quality reads to use for the assembly. So no, I'd say limitation is probably around close to what the size of BACs are.
And would you be able to sequence a mixture of 600 base pair amplicons and a 10 kb plasmid in the same run, for example?
Yeah, this is definitely possible. For ease of downstream analysis, you may want to prepare significantly different size libraries with different barcodes, so you can, for ease of demultiplexing, and so you can provide each barcode folder as input for the workflow separately.
Thank you. Moving to you, Sarah. When we talk about the real-time data analysis, can the Epitome software give us sequence data even when the sequence run is running actively?
So currently the clone validation workflow isn't set up to do that, but we do have some other workflows in the Epitome infrastructure that do support that. And if it's of interest to the community, then it's something that we could think about adding, certainly.
Perfect, thank you. This was also related to Epitome. So can Epitome facilitate alignment to a desired plasmid sequence, or does it need to create de novo sequences? And along that line, can it report if a sample has a mix of different kinds of plasmids within it?
So currently the workflow only does de novo assembly. If there is an interest for a reference-based mode, then if we raise it in the community, then that's something we could consider adding. And as mentioned before, unfortunately, it's not set up to detect contaminants or if there's multiple plasmids. But if we were to add a reference-based mode, it would obviously be easy for us to add a variant calling step, which would help to detect that kind of contamination. So, yeah.
And so the next question, of great relevance, especially to the biopharma and biotech communities, can Epitome plasmid workflow be run entirely offline?
Yes, so you would first need to download and install the workflow either via the application, the Epitome application, or to your command line. And then once it's downloaded and installed, you can go offline and the workflow should run fine on your laptop or device.
Perfect, thank you. Also another question on the different depths of coverage. Is there a lower limit of coverage required to assemble a plasmid construct?
Yeah, so the lowest coverage that's requested or required by the assembler tool we use, which is Fly, as Stephen mentioned, is 10x. But I would suggest using at least 30x if you can, just to make sure that you're getting a robust assembly each time.
Great, thank you. Aaron, maybe you can take the next one. May I use the kits for metagenomics plasmid sequencing, and will Epitome support that approach in the future?
I can take that one. So the workflow we described in this webinar is sort of suited more for assembly, de novo assembly. There is a separate workflow called workflow metagenomics in Epitome that we do provide, and that does allow for taxonomic classification of sequence reads. By default, this workflow uses 16S and 18S NCBI rRNA databases that will be downloaded at the start of analysis. So that's a workflow you may want to consider for metagenomic studies.
Thank you. When it comes to the quality of the samples, what level of QC and normalization is required for input DNA? Do customers need to normalize by raw quantitation or by molar concentration of plasmids with different sizes?
This can help getting an even read distribution across each of the barcodes, especially of different sizes. In general, I wouldn't recommend deviating from our input recommendations for the protocol, and just go with the input recommendations as described in the protocol. And if you need a larger number of reads for your analysis, I would just sequence for longer, have the sequencing experiment run for longer.
Thank you. And Sarah, do we need a reference sequence for each plasmid when you're sequencing them?
So you don't need a reference for each plasmid because we're doing de novo assembly. You can, if you want to, supply a reference for the insert. And if you do that, then a few extra quality control steps will be done at the end of the workflow, just comparing that insert with what was assembled.
Thank you. Jan, the next question is for you. When multiplexing short and very long plasmids, is there a risk to get low coverage of the large plasmids and thus the failure of sequencing verification?
There shouldn't be a big difference. So also difference in terms of long and short plasmid, but also user can visualize in real time in MinNode during sequencing on the coverage, so that we also have a barcode balancing option, so that you can get relatively equal coverage across different size plasmid, and then stop the run. Yeah.
Thank you. Sarah, this question goes to you. Is the accuracy high enough to identify SNPs via nanopore in case of a 10 kb plasmid, for example?
Yeah, so it should be. So you can, as mentioned before, if you want to look for SNPs, the only way to do that via the workflow is if you supply an insert reference, which will then be used to create a VCF report. And yeah, it should be good enough to do that.
Thank you. And we have time for just one last question. I'll move on to this one. Can the workflow be used the same way for large BACs or bacterial artificial chromosomes? Sarah, would you like to take that one?
Yeah, so that's a good question. Unfortunately, the workflow hasn't been tested with BACs yet, and I think there are a few tweaks that we need to make to it for it to be to make sure that it does work with genomes of that size. But it is something that we know that there's an interest in the community about or in. So if there is interest, then we would look to add it in the future.
Thank you very much. And that concludes, we're almost out of time, concludes our Q&A session. And with that, we want to thank you all again for joining this webinar. Please feel free to reach out to us anytime if you have any further questions. We'd be happy to answer them for you.
Thank you all. Now we will close the session here, and I would like to thank you all participants and speakers for your interest and collaboration in this webinar, and hope you have enjoyed and found it useful. If there is additional questions, please reach out to us, and we will be very glad to help you and to answer you. Please do not hesitate to contact your Avantor Life Science specialist if you need more information and support about this topic. Thank you and have a nice day, and see you next webinar.